I'm going to tell you just a little about the human microbiome in general, and it's a fairly new research field, and how it's likely relevant in cancer and cancer therapies. So the human body is really an ecosystem, and our different habitats in our body host different types of microbes. And your gut is where most of our microbes that we have live, and most of them are bacteria. And so in your body, about 3 pounds of your weight is actually microbes, primarily bacteria, which weighs about as much as your brain. Okay? So is it as important? So your microbes, the bacteria in your body actually account for 99% of your unique genes. So if you think about the last talk and how important your genome is, well, this is what we call, there's actually a biotech company called Second Genome that does microbiome research. And so you have this extra set of genes, and these do really important tasks. They train your immune system. They synthesize vitamins that your body can't synthesize yourself with your own genes. And what a lot of them do is they help us break down our food into nutrients that can be absorbed by the body. And so one really unique thing about this second genome is that, unlike your own genome, it can be modified by changing the microbes in your body, and this is where the therapeutic applications come into play. And so just some examples about how important your microbiome can be. Real quick, I'm just going to show a few. So the first and probably the most well-known is the use of fecal transplants to treat recurrent Clostridium difficile infections. And so a fecal transplant is when you take the fecal material, which contains a lot of microbes, from a healthy person, and one way or the other, put it into a sick person. And so in this publication that came out in 2013, they had two groups of patients with recurrent difficile, one group treated with fecal transplants, and one group treated with vancomycin, which is a common antibiotic. And the C. diff infections were so successfully treated by fecal transplant, they actually had to stop the trial and treat everybody with fecal transplant. It's almost 90% effective. And so it's incredibly effective, and this is a really great example of the tool of the human microbiome and the genes and the microbes in your body for human health. Whoops. And so another area of research is, since bacterial really important in how you absorb nutrients, there's a lot of research in obesity. And in this paper, they not only showed in mice that you can modulate obesity with microbes, you can also protect against obesity with certain microbes, which obviously has some therapeutic applications. One of the really hot areas right now is what they call the gut-brain axis. And so this paper here in an animal model showed that you could, with an animal model with the autism spectrum disorder, they showed that you can use microbes to help alleviate some of the neurological symptoms, and this is now an area that's being studied in humans. And important for the talk today, another major area of research is the role of microbes in your microbiome, both in causing cancer, but also in how you respond to cancer therapies. So, in this really, in the response to cancer therapy, this really falls under the umbrella of precision medicine, which was talked about in the last talk. Because, like I said, one of the really unique things is you can modulate the human microbiome by changing the bacteria, and so it's a way that, if you can figure out that certain microbes help you metabolize a drug in a better way, that becomes a target for trying to modify your microbiome. Okay, so what microbes are in the gut? So here I'm showing you the tree of life, and this includes 3 domains. This may have changed since some of you went to college. There's 3 domains of life, not 5 kingdoms: bacteria, archaea, and eukarya. And we're in eukarya, but there's also a lot of microbes in eukarya. That includes fungi, for example. some fungi. Archaea are like extreme environment microbes. And then bacteria, which is what most of us are familiar with, and bacteria is what makes up most of our human microbiome. And specifically, there's two phyla, or two major groups of bacteria that in most people make up about 90% of their microbiome. And just to clarify really quick, because I forgot to clarify before, I use microbes and microbiome somewhat interchangeably, but the microbiome are those genes that are provided by the microbes in your body. So just to clarify that really quick. So the microbes are firmicutes. We have a lot of firmicutes and bacteroidetes. Some of the firmicutes that might be familiar to you from your probiotic yogurt are lactobacillus is a good example. So there's a lot of good firmicutes. And then bacteroides, here I show an example of a bacteroidetes named Bacteroides, which helps break down sugar. But there's, in your gut, you have, depending on how you define species, like a couple thousand or so. So there's lots and lots of taxa. Two of them fall in these really big, broad groups, but there's also other broad groups that include that you have taxa in these really broad groups, but they're in much lower abundance. And one of them that's important is proteobacteria. So proteobacteria is a really large group, and it contains some of recognizable pathogens, such as E. coli and salmonella. It also includes non-pathogenic taxa, but in general, when people have a high level of proteobacteria in the gut microbiome, it's often associated with a disease state. And so how do we sample gut microbes? In a very convenient way. So fecal material does have a fairly good representation of the microbes in your distal colon. They come out with you when you go to the bathroom. And so often in a lot of our projects, if it's a project on humans anyway, we just ask people to swab their toilet paper, and then we extract the DNA from the fecal material, and that includes all kinds of DNA. And then basically what we do is we sequence a gene that's present in all bacteria, if that's what we're interested in looking at, which is usually what we are. So we target the 16S rRNA gene. And if we want to look at other microbes, such as fungi, there's other genes that we target. And then we use the Illumina sequencing technology that was mentioned a bit in the last talk. And this has really revolutionized our ability to study the microbiome, because the limitation before was that you can't culture most bacteria. And then there's so many different species that you needed a large amount of sequence data, a really large depth of sequence data. You needed lots of sequences per sample to really get a good picture of the diversity in any microbial community, whether it was in your gut or in your soil. And so this technology in the last 5 years has really allowed this field to really just explode. And just as importantly is the bioinformatics software, and that's Rob Knight, whose lab I work in. Really, his major contribution to this field has been developing the software that allows us to quickly analyze these sequences as they come off the machine. We have thousands of samples that have millions to tens of millions of sequences each. And we need efficient ways with our various clusters and supercomputers to make this into some interpretable result. And so that's what's really sort of helped launch this field recently. So I'm going to talk a little bit about two different diversity metrics today. So I'm just going to tell you what they are really briefly. The first one is alpha diversity, and that's just the number of types of bacteria or microbes in a community or in a sample. So this person here on the left, has more types of microbes than this person, and so has a higher alpha diversity. And so that's just the first type of metric that we'll discuss. And the second is beta diversity, and that's how similar two communities are. And so here I show example of the oral or mouth microbes versus the gut microbes. So two people, the person sitting next to you has more similar gut microbes than your gut microbes are to your oral microbes. This makes sense, right? We expect your mouth microbes to be fairly different than your gut microbes are doing very different things, right? And so that's the second sort of diversity statistic that we talk about a lot. And so when people are sick, sometimes they have what we call a dysbiosis or an imbalance in their gut. And so this imbalance can happen both with acute illnesses, like if you have food poisoning and you have a big growth of E. coli or something like that. But this is also can be a pattern that we see with chronic illnesses such as colitis. And we usually see certain patterns in alpha diversity and beta diversity in general, and this becomes important in cancer as well, with disease. Okay, so the first very sort of common pattern we see across the board, that's a red flag, is a major decrease in alpha diversity. So there was a recent paper that came out looking at burn victims in the ICU, and even before they've had antibiotics or anything, you see this huge crash in their alpha diversity, which one of the consequences of that is that your intestine can become more permeable, which can be a player in sepsis. So, you know, this is something that's sort of a very common pattern that we're seeing across the board as more and more data sets become available with disease related to the gut microbiome. And a few other really broad sort of patterns that we see is, as I mentioned before, we often see increases in proteobacteria. And it often includes Enterobacteriaceae, which is the class that includes things like E. coli. And then second, we see decreases in sort of the good bacteria. One really important one is Faecalibacterium and also Ruminococcus. And we often see those disappear and go down. And so with the microbiome in cancer, the most well-studied system so far is colorectal cancer, which, you know, is sort of an obvious place to start, that your gut microbiome, where most of your bacteria is, if it's influencing any type of cancer, colorectal cancer is sort of an obvious place to start. And so I'm just going to show you a few slides of sort of where that kind of research is right now. And so first of all, one of the first papers that came out on this sort of showed the documentation of what we expect. Colorectal cancer patients have lower alpha diversity, and they also have a very different community. And one of the sort of trends that's coming out, even though we like to study the whole community, sometimes there's obvious certain taxa that are important players, and one that's becoming clear as an important player is one called Fusobacterium nucleatum, which interestingly is something that's common in your mouth, but when it can get into your gut, and then in colorectal cancer patients, at least, it becomes a problem, and it's been shown in mouse models to help promote tumor growth in a couple different ways, including the Wnt pathway and also by messing with your immune system, because bacteria can communicate and send out signals and mess up signaling. And so that's one of the ways that bacteria or certain bacteria or communities can be potentially a player in tumor growth or causing cancer. And a paper that just came out recently showed that, in fact, when you look at the proportion of Fusobacterium nucleatum that patients have, colorectal patients have in their gut, it is significantly linked to mortality. So so in this case, they're looking at it as sort of a diagnostic, prognostic biomarker, but obviously there will certainly be research in looking at it as an option for targeted therapy, right? And so that's kind of where the colorectal cancer research is right now. There's a lot going on that isn't published that sort of is building on all those types of research. And so it's reasonable to suspect that since we have a lot of microbes in our gut, we see there's mounting evidence that the microbiome is important in colorectal cancer. it's very possible and worth investigating the potential role of microbes in your microbiome in appendix cancer. And so I recently joined this group that had been looking at microbes in the tumor material of PMP patients. This group has published 2 papers, and then this first one, what they looked at was the tumor material, both the mucin and the more cellular material, which looked very similar, and they found that there's this kind of core set of microbes that's always present in the material. And this includes one particularly notable taxa, Helicobacter pylori. So here I show these are the bacteria that they found. These are actually infecting the tumor material, with Helicobacter being sort of the 6th or 7th most abundant one. And as you can see, sort of this trend of most of these taxa fall under proteobacteria, which again are usually either more pathogenic or environmental. They're not really usual gut taxa. And so Helicobacter pylori is known to play a role in other gastric diseases, which I list here, and that includes one type of gastric cancer that, when caught early and treated with antibiotics, can be curable. And so, building on sort of that information, this group that I've recently joined to do further research with also tested the H. pylori antibiotic treatment on some of their patients to see if it had any effect on both the tumor burden of the bacterial burden on the tumor, as well as on survivability. And here I show a slide where the tumor material has been stained, and you can see the bacteria in a patient that was not treated with antibiotics before HIPEC, and a patient that was treated with antibiotics before HIPEC. And you can see there's a lot less bacteria, although notably the antibiotic treatment did not get rid of all bacteria, but it got rid of most of it. And so they also followed the patients for several years, and this here shows the overall survival, and this was after 4.5 years. They had 6 patients who were lymph node negative with the PMC, they used the PMCA DPAM pathology, and compared it to a control group of the same pathology that had no antibiotics. And obviously this is a really small sample size, and so it's suggestive, but the sample size is not big enough to really be statistically rigorous. And so what this group is doing now is they have a clinical trial where they're working with a larger patient group to be able to really assess this more rigorously with statistics. And so the questions that I'm interested in coming from the human microbiome side is taking a look at patients to see if my hypothesis is we'll see some similar trends as we see in colorectal cancer. We want to know, are patients' gut microbiomes altered? What is the relationship between the gut microbiome and the tumor microbiome? And I think this is particularly interesting because, obviously the tumor microbiome is coming from somewhere, and it's most likely some selective event from the gut microbes, and so trying to understand how that happens and what the connection is, I think, could be insightful. And then how does the H. pylori antibiotic treatment affect the gut microbiome? And this more goes with sort of quality of life. We know that a lot of people who have had HIPEC have chronic diarrhea issues, and this is another area where microbiome research could probably be useful for helping to improve gut health in general. And so where we're starting is with 24 to look at the gut microbiome of a subset of the patients that are involved in the clinical trial at Mercy Medical Center. Sorry, I made my PowerPoint on a Mac, so the spacing didn't quite work out here. But anyway, So we're going to follow 24 patients at 3 time points. One is at diagnosis before any treatment, and then soon after HIPEC, and then 3 to 6 months later. We're going to look at their gut microbiomes and compare them to between both patients. Half the patients will do this with half patients that are receiving antibiotics and half that are not. And then we're doing this project through a really big citizen science project we have here at UCSD out of the Knight Lab called the American Gut Project. And this is a project, it's done through fundraiser where it's citizen science because you can donate, you can contribute $99 and then we send you a kit, you swab your toilet paper, and we generate your microbiome data. But along with that, there's a big survey that you fill out. And so this has allowed us to build a really huge database of gut samples of people in the US. And so what we'll compare, so we were going to run this gut microbiome PMP patient project through the American Gut, and we'll use an age-matched set of healthy individuals based on their surveys as our comparative group to document the dysbiosis that I'm almost sure will be there.
When does that start?
The American Gut Project? The IRB is being reviewed now, so at Mercy Hospital. And so with the American Gut Project, basically this is how that works. It's as I mentioned, it's this fundraiser. You get a sample, it comes back, and then you get results that are sort of at the broad level showing some of the metrics that I showed you today. But importantly for us, it also provides us with a really big database that we use to compare with other clinical types of projects. And we have one PMP patient that's participated in the American Gut Project, and we have 2 time points. And one thing, so we have an N of 1, which is always a good place to start. And so first we look at the alpha diversity, and we see as expected, the patient has, almost half as much different types of microbes as a healthy person. So the sort of what we would expect with someone who has an illness, particularly one related to the GI tract, is we see this low alpha diversity. And we also see different taxa, and some of these are very similar to what we see in colorectal cancer. So most notably, at one time point, interestingly, at one of the 2 time points, but not at both, the patient had a strikingly much higher abundance of Fusobacterium nucleatum, so the taxa that is implicated in colorectal cancer as a player. And this is a second taxa that's also being studied as a biomarker and potentially having a pro-inflammatory role in colorectal cancer. And at that same time point, had a high amount of this. And then also notably is what's missing. And so Fecalibacterium is a really important gut microbe to have for healthy epithelial cells, and you can see that. So these are there's different scales here, but on this scale, most people have 10 to 20% of your gut microbes are this taxa, and it's really important. And here, it's almost not detectable in PMP patients. So this is why we need to document that there is a dysbiosis, but I suspect that we will see a dysbiosis both because we have this fantastic N of 1, but also because it makes sense that we'll probably see a problem. So in conclusion, this is obviously just getting going with appendix cancer patients, but we think that it's very likely there'll be a dysbiosis, and there's probably a lot of different avenues that we may want to investigate to improve patient health. And beyond the 3 questions that I stated earlier, there's a couple others. I think it would be interesting to see, similar to colorectal cancer, if there are some associations with the gut microbial communities and something like pathology or mortality. And then sort of as I alluded to before, I think there's probably some, it's sort of a separate kind of thing, but I think that there's probably some opportunities to try to improve gut health in patients by studying the microbiome and potentially looking at either diet or probiotic interventions. Although this has to be done carefully, there was just a paper published showing that in mice, so one probiotic that's very good for many things, VSL number 3, was shown, and it has been shown to help prevent colorectal cancer in mice. But once mice have colorectal cancer, it actually helped the colorectal tumors grow. So we have to be careful in really studying the effects of, because like I said, you're changing what accounts for 99% of your unique genes in your body when you mess with your microbes. So, it has to be approached carefully and systematically with research cohorts. And if you're interested in learning more about the human microbiome, we have an open online course through the Coursera platform. It's sort of at the very broad level for the public. It was mainly made for participants in the American Gut Project. And yeah, so that's free online, and you can find it on Coursera.org. You can just search for microbiome. And that's me, one other postdoc, and Rob Knight. And then finally, if you're interested in the American Gut Project, you can also come talk to me, but it's also easy to find online at american.gut.org. And like I said, that's a project that's actually here at UCSD. And I'd like to acknowledge the many people who have spearheaded this work. As I mentioned, I've just joined fairly recently to try to add the gut microbiome component. And so I want to thank all those people and their funders, and then also the American Gut Project team, which is sort of where we'll work through this project through. And so I'm happy to take any questions. And I have a personal investment in this, so.
Jessica, that was great. I'm going to take the privilege of a first question
Sure.
to ask you. So obviously you mentioned the open question about how cancer treatment can affect the microbiome that we don't know. I assume that lots of other things, folks take antibiotics, they get antibiotics when they have an operation, et cetera. So in approaching this and gathering data from patients are in very different places with respect to their treatment course and their disease course. And so how are you planning to account for that and the differences that you might see just on the basis of treatment-related, disease-related factors?
That's a good question. So I think with the project we're starting with, we're looking for such broad trends. So what I expect to happen is that at diagnosis, you already have a low alpha diversity in the patients, and through treatment, it's going to crash and crash further. And so that might be something to target just for GI health in some way. But first, we have to document the dysbiosis. And in that one, we'll have 3 time points where the patients are, within 2 groups have been given more or less the same treatment. But there certainly will be variability. And that's the real challenge with the microbiome is our human genes are 99-point-something% similar, but your microbial genes, day to day, are more variable than that, and person to person are extremely variable. So although you have this opportunity to modulate it, it's super hard to study because of the variation, but that's why you need these big bioinformatic platforms. But I think the key will also be lots of patients in the end, but we don't have funding for that right now. So.
Great. Sure there are other questions. Yes.
I've been reading a lot about this, and what you eat can change your microbiome within 24 hours is some of the information that I've received. Has there been any study where you look at the microbiome in terms of pre-HIPEC, post-HIPEC? I imagine HIPEC probably wipes out most of the microbiome in a patient. And maybe that's part of the magic.
Well, so that's what we'll be able to do with this, these 24 patients and the 3 time points. So far there's only one data,
Right.
2 time points, but one patient that we've looked at, and those were both post-HIPEC. So we have no pre-HIPEC data points at all right now. So.
Would that be a hard study, expense-wise or any other way?
So that cost is, cost and then organizing the IRB type of stuff is always the issue. What's really nice about studying the gut microbiome is that it's not invasive, so it usually is easier to get permission from it. That's partly why we also have the American Gut Project, so people can choose themselves to participate, and in your survey, you can put pseudomyxoma or appendix cancer, and then you become part of a database that when there's enough people, so even if you're not part of a particular study, you can still get your gut microbiome data and be able to compare it. When we give results, you get compared to other people your age with your similar diet, and then to Michael Pollan, who's...
One other question.
Yeah.
I've heard studies or discussion about, 80% of your immune system is controlled by the bacteria in your gut.
Yeah.
Something like that. Is there any studies regarding the immunology and microbiome?
Yeah, so that's a really important area of research, is how the microbiome affects the immune system and vice versa. And certainly the window of time that's most important is your first two to 3 years of life. And there's a lot of research on this in autoimmune diseases. So there's a lot of pretty compelling evidence that sort of having a lack of microbial exposure increases the incidence of certain autoimmune diseases. And so in that sense, like training the immune system, it's really important. And that's really, our understanding right now is that the most important window is in the early part of life. But certainly there are ongoing interactions. So, with cancer, one of the things that's being studied that's really exciting is sort of like when you have something like H. pylori, what it's probably doing is there's this little microenvironment around it, and it's modulating it. It can communicate with different signaling pathways or with your T-regs immune cell. So in that sense, it can, in a very sort of microenvironment, interact with your immune system. But there's a lot we don't know, and it's we're just starting to try to understand it, and it's very challenging because it's so complex.
We're running a little behind, so I think we're going to break now. I'm sure Jessica would love to, if she'll be around to answer more questions.